All of these are zoonotic from animal to human viruses. And when they're often, we've always been afraid there would be another pandemic worldwide. Usually most virologists thought it was going to be caused by flu viruses, but we should have taken more caution when we saw what happened with the SARS and the MERS. Let me just say that the case fatality rate in influenza is about 0.1%. I've gone through some of the COVID-19 case fatality rate in the range of 3%. SARS that in 2002 had a mortality rate of about near 10%, and MERS has a mortality rate of about 33 or 34%. Incubation times 1 to 4 days for influenza, 4 to 14 days for COVID-19, 2 to 7 days for the SARS, and about 6 days for MERS. And it just shows that the infections rate, the ability to infect others, was low in MERS, probably was high in SARS, but was well controlled. And in this one, we have an infection rate, the R0 is high enough that we have had trouble controlling this virus. Of course, the approaches to control a new virus outbreak is the public health measures that you're all aware of and lived through: closure of public events, social distancing, hand washing, testing and tracing, and lockdown. Antivirals are very important, and there's over 100 in studies and well over 200 clinical trials that are currently going on. And vaccines, there are about 139 vaccines in preclinical studies and about 30 in clinical trial phases. You mentioned that I was on the vaccine task force. I just have here the members of the vaccine task force, and some of them you'll clearly recognize, but our 2 deputy ministers of health and of innovation, science and economic development, Stephen Lucas and Simon Kennedy, are on this committee as well. We report and are advisory to the deputy ministers of health and to these 2 ministries and to the government of Canada. In the end, we want to find vaccines that are safe, effective, and we want to make sure Canada has timely access to these vaccines. Countries are building bubbles around them. Access particularly important. The Canadian government will do the procurement agreements. We don't have anything to do with that. We just give advice, and the vaccines must pass the health protection standards and be licensed before they can be used. Worried about making sure that we get this vaccine out to as many people as we can when it is available. Vaccines are different than antivirals. Vaccines are for large populations of healthy people. Antivirals are directed at people with the disease. So there is more tolerance for the side effects, I think, in antivirals, because you're looking at a balance of safety and benefits. But for vaccines, global mass immunization, vaccine safety is of absolute paramount importance. And I just want to assure the public and the people as much as possible. At the present time, there are new platforms so we can get vaccines developed faster. Of course, with molecular biology and new technology and science, you would see some of these coming through much quicker. And we've seen this with inactivated vaccines, live vaccines, vectored vaccines, and this is where the new ones come in: the vectored, the adenovirus, and the VSV vectored, messenger RNA, self-replicating RNA, protein subunit vaccines, virus-like particles, and meant to have DNA vaccines on here as well. I just want to assure you that Canada and the Vaccine Task Force, we had our first meeting on June the 16th, and we've had 36 meetings since that time, all varying between 3 and 5 hour Zooms. So the Vaccine Task Force has been very busy, and the federal government has made procurements. with different vaccine companies. Moderna, 56 million, Pfizer, 20 million. I think they've just increased that. Novavax is 76 million. J&J has got sorry, it was 38 million. And so Sanofi Pasteur just announced yesterday about 76 million. This sounds like a lot of doses. None of these are come into effect until they're proven safe and licensed. And many of these will require 2 vaccinations, so that you've looked at the number of Canadians. We've got, the government has been active in trying to make sure we get access to good vaccines. So if vaccines are not effective, what does that mean? Well, I just want to point out that there are 2 diseases in the world where we have never been able to develop a vaccine. HIV/AIDS, but we have excellent long-term therapy, and hepatitis C virus, where we have no vaccine, yet we can have 95% cure. So yes, the work on antivirals is critically important. We don't know how effective and how long our vaccines immunity will last. And I just want to emphasize that antivirals are a very important part that we need to follow up. I want to say one other thing as we go into this, that once you're infected, there's an incubation period, there's a respiratory phase. Many people get better, never have a disease, but some will go on to respiratory, and then others will go on to multi-organ immunopathogenic phase of their disease. And this is a short course of about anywhere from 10 days to 30 days. Some people are in hospital much longer in this long-term immunological phase. But we've talked about changing, flattening the curve to prevent people getting overloading the hospitals. And I think we did a pretty good job of that. And we're looking at the second phase, and we'll have to look at it again. But there is another issue, is changing the course of the disease. And in a short disease, if you're going to use antivirals, I think you're going to have to use them fairly early in this disease pattern, and not when you get out to the very severe patients that are in ICU. That's going to be a tough place for the antivirals to work. I'm not going to go through this slide in any, but I just want to show you that this is a disease that you don't want to get. It does have the potential for multi-organ disease, cardiovascular, central nervous system, gastro, kidney, many different immune complex diseases. So it is a bad disease, and that's why we emphasize so much about getting, preventing the disease as much as possible. There's lots of sites within the virus that we could look at antiviral agents and potential of these antivirals to inhibit. And we're going to be talking today primarily on the proteases of this COVID-19 or the SARS-CoV-2 virus. And it is one of the main targets. Other targets have been the polymerase, which has been the major target for a drug called remdesivir. And the thing that really precipitated a lot of the interest in the antivirals was very early on. A paper was published in Nature that said, showed that remdesivir, a drug that was originally developed for Ebola, had good antiviral effects with a low amount of virus inhibiting the virus. Selective index was excellent, so we thought this was going to be a very good drug. The other one was chloroquine, and it also showed some benefit. I just want to say that remdesivir did not affect the entry. If you look at here, if you have the virus there, full time, the antiviral there suppresses it. But if you use it to try to block entry, it doesn't do anything. But after the entry, it has effect on the virus and drops the virus dramatically. Chloroquine, on the other hand, focuses mainly on entry. And after the virus is in the cell, it doesn't do much. Let me say that I'm not going to talk about chloroquine anymore and hydroxychloroquine because they have been tested and they really not shown great effectiveness. There are only 2 drugs that have been licensed. And most important is remdesivir, which has a structure similar to ATP. And there has been excellent work done at the University of Alberta, as well as other places. But I think some of the leading work on remdesivir has come out of Dr. Matthias Grote's lab at the University of Alberta, and 2 key papers that have come out really outlining that it does work against the coronaviruses and that it mechanism of action. Let me just say that his first paper showed that the virus had a good effect against MERS virus and with a low effective dose 50. And the second paper, which clearly showed that this virus inhibited the RNA synthesis, this antiviral inhibited the RNA synthesis, does it in a rather unique way. It gets inserted into the RNA, matching up with uridine, and goes 3 nucleotides downstream. If you have, and that's where the termination occurs. It's a unique property because remdesivir triphosphate is highly selective to being incorporated into the RNA, and even more selective or more preferred than the natural nucleoside template or ATP. It is selected. However, when remdesivir was first tested in populations, it did not look very much better. If you look at the cumulative improvements on the red line and the blue line that didn't get remdesivir. However, these studies were followed up, and their publication in July in the treatment of COVID-19 with remdesivir. And I would just say that there has been definite improvement, and patients improved 31% faster compared to the patients who received placebo. So in many parts of the world, we would consider remdesivir part of the standard of care. I would say that it's very important, again, that you try to get into these, get into the patient early rather than late. And the problem right now is still access to remdesivir. The second drug that has been approved is dexamethasone for the treatment, and excellent studies that have been done. They've been reported in New England Journal of Medicine. And let me just say that these curves show that the patients receiving dexamethasone have about a 30% less death rate and are improved and have less oxygen, less number of patients going on to oxygen. And dexamethasone is absolutely one of the standards of care for the treatment of patients, particularly when they get into late disease. Let me just say that once again that, infection, incubation, respiratory phase, this is where we would like to use the antivirals like remdesivir or other new antivirals that might be tested. The dexamethasone has an effect even in this late phase, and that's where it is distinguished from the other currently antivirals. Only the 2 have been licensed, to my knowledge, at the present time for the treatment of COVID-19. Let me just say that in antivirals, we can be thankful for carefully conducted clinical trials. We saw what happened when political interference got into the anti-malarial drugs and how that altered the impression. It caused a lot of confusion. The only way to solve research problems is major challenges is through research. Basic research is critically important to healthcare, and this is what will transform how we handle this disease. Teamwork and collaboration is essential ingredients, and I don't think we've ever seen such good teamwork and rapid collaboration, both nationally and internationally and locally, as we've seen with the COVID-19. I also want to acknowledge the rapid response funding that we received through CIHR. It's critical for the laboratories that we're trying to switch and get into COVID research. I feel badly for those others that are not in COVID research because it has been an advantage for our labs to receive this funding. Let me just say that antivirals for new diseases often take time. And I mentioned there's no vaccine for hepatitis C, but it took a long time to develop the effective antivirals for hepatitis C. The virus was discovered in '89, and the most highly effective antivirals were 2014, 2015. AIDS discovered in 1980, and we got, it took a long time, again, almost decades in this case, years in the case of HIV AIDS, and in SARS, COVID-2, we're looking at having antivirals. We've got 2 already, but we're having antivirals in months to a relatively short period. Tremendous amount of research, tremendous new technologies that will allow us to develop antivirals for this virus faster. So the main target that we have worked on with Joanne Lemieux and John Veteris and the teams in all of our labs has been on the main protease, a key protease that cleaves the polyprotein into its various components that make up the virus. I'm going to turn the screen over to Joanne Lemieux, who will discuss our work on the feline COVID virus and the antiviral agents. Thank you.
So thank you, Joel. I'm just going to share my screen. Okay. So I'm very happy today to talk about our work that we've done as a result of, as Dr. Tyrrell said, very strong teamwork. And this, sorry, I have to escape. This was recently published in Nature Communications. So as Lauren mentioned, we have great teamwork in this project. And I want to emphasize that this grant was funded at the end of February, and we submitted this paper at the end of April. So during the time in March and April where most labs were shut down and most people were hunkered down at home, our lab members were all in the lab 7 days a week, really doing a tremendous amount of work. And the collaboration here at the University of Alberta between the Young Lab, the Tyrrell Lab, Veteris Lab, and my lab was really quite phenomenal. Okay, so as mentioned by Dr. Tyrrell, we're focused on a feline drug that is known to target the protease in the SARS coronavirus. And so what I have here is a diagram showing the single polypeptide chain that is made by the coronavirus. So the SARS-CoV-2 genome is about 30 kilobases, and all of these, this one long polypeptide must be chopped up into pieces in order to generate both the non-structural proteins, so these mostly play a role in replicating the nucleic acid of the virus, and then we have the structural proteins. And so the role of the proteases really is to chop this long polypeptide up. And the main protease, or the 3CL protease, cuts the polypeptide at the gray, light gray triangles. There is another protease in the coronavirus genome, the PL protease, which cuts at the black triangles. But I'm going to focus mostly on the 3CL protease. And you can see there are 11 sites that this protease cuts in the polypeptide. And this really demonstrates the essential nature for this protease in generating virus in cells. So I want to discuss first why proteases are suitable drug targets. So we know that proteases cleave a substrate. So here is an example, just an illustration of a substrate. And we know that proteases recognize specific sites in the protease. And these subsites in the protease will dock the substrate and allow cleavage to occur between a bond. And so this bond is called a scissile bond. And the nomenclature that's been ascribed to it is the P1 position and the P1 position. And you can see these positions extend from there up to the P2. And if we go backwards internal to this, it's P2, P3, and P4 position. And this very precise recognition makes proteases excellent drug targets. And importantly, it has to be considered whether or not similar proteases exist in our system. So the main protease that's found in the coronavirus, we do not have an analogous protease found in human systems, although we have ones of similar class. So just to, I want to bring up other protease inhibitors that have been developed to use in clinics. And so many of us are familiar with these. So we know that there are inhibitors that work against ACE and renin. and they are effective in treating cardiovascular disease. And we also have protease inhibitors that are used to treat diabetes, and they're listed here. Dr. Tyrrell just highlighted the different proteases inhibitors that are used to combat HIV as well as hepatitis C virus. So these really have made great strides in combating viruses where vaccines could not be developed. So this really does tell us that developing drugs against the protease is a valid approach. Now, I want to bring some history into this. So this is not a new issue that we've dealt with here at the University of Alberta. So back in 2003, when we had the initial SARS outbreak, the labs of John Vetters in the Department of Chemistry and Michael James in the Department of Biochemistry developed an inhibitor against the main protease for the SARS outbreak. So this is the 2002-2003. And I happened to be a postdoctoral fellow in Michael James' lab at the time and was aware of this work. And because of this precise recognition of the SARS-CoV main protease, they could develop an inhibitor that was effective in inhibiting the protease. And so this was published in 2007. And what this allowed them to do is to develop an inhibitor that had groups that mimicked the binding site. So we know with the SARS main protease, that P1 position I described at the beginning is typically a glutamine. And so because of this, they could insert a glutamine surrogate. So this is a gamma lactam group, and this could be recognized in this region. In the P2 position is a leucine, and the P3 position has been quite open. And so in doing so, they developed an inhibitor that was peptide-based, a peptidomimetic that mimicked the precise cleavage site. And then they were able to solve the three-dimensional structure of this inhibitor. So the inhibitor is in light gray, and you can see dashed bonds that represent hydrogen bonding networks. However, we know at this time that public health measures did really quell the SARS outbreak in 2003. And this research really wasn't pursued further in these labs. However, the groups of Pedersen, Kim, and Grautas and Chang at the Kansas State University decided to take advantage of this knowledge or use this knowledge to really understand how this inhibitor worked and possibly apply it in veterinary cases. And so essentially they used a very similar backbone substrate. However, they're just missing this end piece, and they have a different leaving group here. And what they were able to show is that, and so I'm skipping over years of work here, trying to quickly summarize it. They were able to show that this drug was effective in reversing the progression of a fatal form of coronavirus infection in cats. And this infection is called feline infectious peritonitis. And I kind of want to bring up what this is, because I think it's really quite important of how this drug was used to treat cats, and then whether or not it can be transformed and used in humans. So we know cats can be infected with a form of coronavirus, so FCoV, and this is a non-harmful form. It typically results in mild respiratory symptoms. However, this virus can mutate at certain points within the genome to cause a more invasive form of the virus, and this leads to the feline infectious peritonitis. And in cats, this is almost always fatal. It has a big problem with litters, with kittens. If there's an infection, the whole litter can be infected. And this is a serious problem because many of us do have pets and they're part of our family. And you know, it's really nice to have medicines developed to treat our feline friends. So let's discuss how the drug was tested. So in this study, they did very detailed pharmacokinetics to demonstrate that the drug did stay in the bloodstream for at least 48 hours. They did a 4-week safety study, and then they went on to do an experimental infection in cats. And to do this, they pre-infected cats, either in the peritoneal cavity or in the ascites, and they monitored treatments and they monitored symptoms, and upon the emergence of symptoms, they treated the cat with the drug, with this feline antiviral. And they found in the peritoneal cavity, 4 of the 6 were able to recover. With the ascites administered virus, the drug was able to reverse the virus progression in 2 out of the 4. So in summary, in their paper, they really showed that this was an antiviral treatment that could significantly reduce the viral loads in the cats, and it was an effective treatment for feline infectious peritonitis. So this drug has now been taken on by a company called Antivirals, and I have a few slides at the end to discuss this company. So this company is a veterinary medicine company. Now, some people may ask, cats, why would I take a drug that has been developed for cats? I just want to emphasize that there's some other drugs that humans take that you may be aware of that were first used in animals, and now they're used by humans. So ivermectins are an antiparasitic that were first used in animals. And then we also have cyclosporins, which were first approved for use in dogs. So as Dr. Tyrrell did mention too, so if we piggyback some medicines that have been approved in animals, it does allow us to speed up the development of drugs that can be effectively used. And this is quite important in a case where we're dealing with the pandemic of COVID-19. So now I'd like to start to discuss the drug that was used in the feline study. So the name of the drug currently is just numbers and letters, GC373. And this drug has been studied, as I mentioned earlier, quite a bit with different proteases, so from various viruses. So it was shown to be effective against picornavirus, norovirus, so this is a virus that would cause the stomach flu, and various coronavirus, so SARS, MERS. And so this team really has shown already that this is a very nice broad spectrum antiviral. And while we're discussing here the possibility for use against COVID-19, it possibly could be used for other purposes. Now, my lab is a structural biology lab, so we are interested in understanding how this drug works at the mechanistic or the molecular level. And prior to us working on this, there were 5 structures deposited in a database that holds 3-dimensional structures of proteins. And upon us starting to work on this, we now have evolved to having over 20 structures. And so it really shows that we were working in a very competitive field, and we were lucky to slip in down here and get this work published. A little bit more in the background of this drug and more work in animal models. So this group also recently showed that this inhibitor is effective to block MERS coronavirus in infected mice. And because of the very close relationship between the SARS and the MERS coronavirus main protease, this again fosters confidence for clinical trials. So on to our paper. So back in beginning of April, we wanted to ask the question, is the feline drug effective towards SARS-CoV-2? So this is the virus associated with COVID-19. So here's the chemistry of the drug. We actually have 2 forms. We have a prodrug. So this is the peptide backbone that's going to be act as the inhibitor. And we have a moiety called a bisulfate group, and this makes the drug very soluble. So it allows us to solubilize the drug at a high concentration so that we can administer this. And actually, I did forget to mention in the feline studies, this was administered it was an IP delivery of drug. And so with this drug, upon dissolving in an aqueous solution, this bisulfate group will leave and you get the final drug. And I'm going to point out that P1, P2, and 3P position because I'm going to get into the molecular aspect of this drug. But here's the active group. And this active group is going to react with the nucleophile or the reactive group, which is a cysteine in the main protease. All of this synthesis of the drug and the prodrug were accomplished by the teamwork in the John Vetters lab in the Department of Chemistry here at the University of Alberta. Now, before we tested the drug, we did a sequence alignment looking at the similarities between the protease of the SARS-CoV-2 with the SARS-CoV-1. And what you can see with these straight lines, it shows they are identical. The single dot shows they're similar, and then they're highly similar with 2 dots. And we only have 4 residues that are really distinctly different. So we were quite confident that this drug would work since it had been shown to be effective against SARS-CoV-1. Starting out with this work, we purified the protein and we did some basic kinetics. This looks at the how fast this protease will cut a substrate that was also generated in the Veteris lab. And so this really is useful because next we wanted to analyze whether or not the drug would inhibit the activity. So we demonstrated the proteases we made were active, and next we wanted to test the IC50, or the inhibitory concentration, or the concentration at 50% to give inhibition. And indeed, both the drug and the prodrug with SARS-CoV-2 was able to inhibit the protease. For comparison value, we compared the SARS-CoV main protease IC50s as well. And these values are in the order of 400 nanomolar to 190 nanomolar for the SARS-CoV-2, and a little bit better for the SARS-CoV-1. So this is an in vitro study. So importantly, we needed to demonstrate that these inhibitors would work with the live virus. And so this work was done with the teamwork in the Tyrrell lab. And so in this study, they have plated cells. These are Vero cells, so they're female kidney monkey, female green monkey kidney cells. And there's a lawn of these cells with a dye. And when the virus will infect these cells, it forms these plaques that can be counted. And with increasing amount of drugs, you can measure how many plaques will form. And you can determine from this the effective concentration that could be useful. And we showed, they showed that both the GC373, so the drug and the prodrug, were effective in inhibiting the live virus. Simultaneously, toxicity studies were done in both Vero and lung epithelial cells. So this is human lung epithelial cells. And from this, they could measure the cytotoxicity, which is greater than 2 micromolar. And the EC50 from this was about 1 micromolar with a therapeutic index of greater than 200. So this really shows that this drug was in the range to be useful for clinical trials. Lastly, the group in the Tyrrell lab also looked at viral RNA that was being produced in the presence of the antiviral inhibitor. So with increasing amount of drug, we detected that a lower amount of RNA was being produced, and this value can be very important in also assessing that the virus is no longer being made and generated upon the administration of this drug. And so my lab then wanted to look at the molecular mechanism behind this. So again, we had the protein. We do X-ray crystallography where we take the protein and precipitate it, and we can use X-ray crystallography to look at the 3-dimensional shape to determine the molecular mechanism of this drug. And here's the overall structure of the protease, and you can see in this little pocket here, and in pink is the drug. So if we take a closer look at this, we can look at the different domains of the protein. So there's 2 beta barrels here from anyone who's interested in the biochemistry of this, and the drug sits nicely between this, and it has a covalent reaction with the sulfur group of the cysteine. Now here I want to show you, I just need to exit my pointer. Here I want to show you a movie about this protease. So this protease behaves as a dimer, and this movie really shows a 3-dimensional shape and structure of how the protease comes together and how the drug works. So what we're doing here is we're kind of zooming into the active site, and I want to point out this region here that we have the active site cysteine with a general base histidine, and this is the reactive group where the inhibitor will bind. But I also want to show that there's some residues from the other protomer, or the other part of the dimer, that interacts with residues near this active site. And so this is going to be quite important for the molecular story of this. Okay, so moving on to the drug and how it reacts. So we crystallized the protein with both the prodrug and the drug, and we were able to show a covalent interaction as we expected. So these were essentially identical, and this forms a hemithioacetal interaction, which was confirmed by NMR. Here is the active site pocket, and again, we see the drug fitting nicely between these two regions. So when we're thinking about drug development, first we want to look at how the current drug is working, and then we want to look at this a little bit more closely so that we can possibly make some adjustments if we want to improve upon this. So here in the next few slides, I'll talk about the molecular interactions, and what we're seeing here in pink is always going to be the drug. In the black hatched line, we see hydrogen bonding network. And we see that there's a lot of regions that hydrogen bond with this drug. So if you recall the P1 and the P3 positions, we see the P1 position is hydrogen bonding with these two residues. And these I will discuss in the next few slides as well. There's also hydrogen bonding with some of the negatively charged regions. So this is also called the oxyanion hole for people who are knowledgeable about membrane or about protease structures. The P2 moiety is nestled in this little hydrophobic pocket between methionine residues and histidine residues. And we can take a closer look at that interaction here. So you can see there's a lot of hydrogen bonding here that occurs. And this hydrogen bonding network is what gives the very high affinity for this drug. But I want to point out these two residues here. So this is the hydroxyl group from a phenylalanine and a glutamine residue. And earlier I introduced that in the dimer, there are residues at this other protomer that interact with these two glutamic acid and hydroxyl group of this phenylalanine. And these are the ones that actually coordinate the drug. So there's a very nice fit of this drug with all of these residues. I will point out that this P3 position is not really hydrogen bonding with anything. So this is obviously an area for optimization. And if we look at the structure of this in a space filling model, you can really see the fit, that we have a good fit with the P1 and the P2 position, but the P3 is kind of sticking out. So our labs are still really working on optimization of these drugs to make better derivatives. And this includes a new collaboration with the Niemann group at the Li Ka-shing Institute for Virology. Moving forward, we've also crystallized the feline drug in the presence of the main protease of the SARS-CoV virus, and we see very similar structures. So if we zoom into how the drug fits in both SARS-CoV-2 and SARS-CoV-1 main protease active site pocket, we see almost identical fits. So this again really supports the notion that this could be a possible broad spectrum antiviral. So with our team, we're doing some inhibitor development, as I mentioned. We do also plan to look at how the drug works with the feline coronavirus, because we think this is quite interesting and it hasn't been done yet. And lastly, I just have a few slides about our collaboration with Antivirals, which is the veterinary medicine company. So they play a role in developing drugs for animals in veterinary medicine to treat fungal infections, lymphoma, as well as feline coronavirus. And so they were well poised when this outbreak took place. And on their website, you can see the update of the progress for clinical trials. So they're at the mid stage. They've completed the IND, so that's the FDA investigation of new drug, and they've done efficacy and safety studies in animals, which are complete. And so here at the University of Alberta, Dr. Tyrrell and co-applicants, Veteris Schwartz and myself, have been funded to proceed with a clinical trial of this here in Alberta. And so that's our talk today, and I'll be happy to take any questions as I turn off my phone that's ringing. Sorry. Oh, and so lastly, I'm not going to give this summary, but lastly, I'd like to acknowledge all of our team members. So in the Department of Chemistry, led by John Veteris's lab, they made all of the drugs and the inhibitors and helped with some of the kinetic assays. In the Department of Biochemistry, Dr. Young's lab and my lab did a lot of crystallography. That would be Bashir and Elena, and we have other team members working on future studies. And then in the Department of MMI, under Lauren Tyrrell's leadership with Justin Unholly and Mike Joyce, and we have a new collaboration with Dr. Neiman. And we have some synchrotron facilities to thank for the crystallography, as well as the funding groups, and especially the rapid response for COVID-19. My lab has worked on proteases for a very long time, and so it really helped us be quite ready when this, this analysis was needed. So thank you.
Great. Thank you so much to both of you. We've had a few questions in the chat window, and maybe we'll start with one of those from Kathy Flood, who asks, is there any evidence that the efficacy of the virus is decreasing worldwide?
I don't think we have enough evidence to say that yet. I imagine by efficacy, do you mean that it's getting less severe or causing less severe disease? I think that, you know, we seen the disease in that first pandemic with a lot of the deaths occurring in people over the age of 70, those with comorbidities. In the second pandemic, I think the death rate appears to be lower, but it is also occurring much more in young, healthy people. And I think that you're going to see changes in the death rates and the effects, depending on the key populations that are targeted by the disease in each pandemic. I don't think that we have enough evidence, but there are studies ongoing, including studies here at the university, monitoring the genome and seeing if you can find genetic changes that might account for some changes in pathogenicity.
Okay, thank you. Maybe one more question. Nancy Liu, who asked, what are some of the challenges that you think you'll face with clinical trials?
Well, one of the challenges that we are facing, and you know, is solubility of the drugs. And the prodrug, GC376, is very soluble. And if you're administering that, and I think the reason that these drugs were all administered intraperitoneally is it's easier to administer intraperitoneally in cats and in animals than it is to go intravenously, and we don't have oral drugs that are well absorbed yet. But there is work going on, and I'm sure John Vetteris and Joanne may comment further on trying to develop drugs that might be orally available, or we would rather in humans use this intravenously. And with the Navi, they were focused on animals, but they're now focusing more on humans. We're doing some studies on the solubility to make sure our drug is very soluble. John Vetteris has demonstrated the G376 is a very soluble form. And when it goes to GC373, it is much less soluble. So we have to work that out, because if you're giving it subcutaneously, you want to be able to use a small volume in patients, even in the phase 1 trial. That's the only thing that's holding up our progress to get to the phase 1 trial here and submit it for pre-CT approval and move on to a phase 1 trial.
Mm-hmm.
Okay.
And I think Lauren, earlier on, we were worried, like in the summer, of not having enough patients.
Yeah.
But that has changed.
Yeah, no, well, you know, in the phase 1, we'll be using healthy people. That's why I was hoping we would get something that would be easy to administer. But in the phase 2 studies, we'll be moving, and there is a promise from Navi to sponsor the phase 2 here in Alberta, and we will, I think, not have the problem with the patients. And we do have a setup across Canada where there is a group working on COVID within Canada, where they can rapidly enroll patients across the country.
Okay. There was a question earlier on from Sylvia Coso, who asked, because ATP is present in so many processes in human cells, How does remdesivir ensure selectiveness to the selectiveness to the virus itself?
Mhm. So I can answer that. So this is a nucleotide analog, and it's really going to just work with RNA synthesis and not with replacing ATP by itself. And because you can imagine a short term of this drug being delivered, it's not going to have effect on other rapidly dividing cells, but it should not complicate ATP in the cell.
Great. Thank you so much. Are there any other questions? Okay, so we have a follow-up question from Kathy Flood, who said that there's a coronavirus vaccine for dogs, and wondering if that would be of use for vaccine development.
Well, I think that there have been some coronavirus vaccines, as you said, developed for dogs, and I think that's encouraging. What we don't know yet about the vaccines is we do see they can induce neutralizing antibodies, and they do introduce or induce neutralizing antibodies in levels comparable to what we see in patients with convalescence after they've been infected with COVID. So we get good titers, and in some of the vaccines, the titers are even higher than we see in the average convalescent sera. I just think that, I just want to emphasize how important safety is in vaccine development. And again, I'm sure we get clues from the dog work, but everything that's been done in vaccines is being done extremely carefully. If you have 1 in 10,000 patients that develop a serious side effect that is unique, that will kill the vaccine. It will not go much beyond. So there's 30,000 patients in most of the phase 3 studies. So you're trying to have enough patients in there that we can ensure vaccine safety. But, we've seen what happened with diseases such as Guillain-Barré with the influenza vaccine that was used in H1N1, killed that vaccine very quickly. And we want to make sure there's no side effects and try to be as safe as possible.
And I think the other thing too, Lauren, is as we've discussed together, that there's still a priority for antivirals because there are some people who may not be able to take a vaccine.
So our big concern right now is not the anti-vaxxers. The anti-vaxxers are fixed in their thoughts that vaccines are not going to be accepted. But there's a big group that are what we call vaccine hesitancy, because these are new vaccines. And I think it's going to be, not only in vaccines, I've learned very clearly, there's what we call a vaccine substance, which is how you make a vaccine, and the university can design great vaccines, but then you have to get a vaccine product. And this is where you involve industry and how do you get a product out, and how do you get into public health, and how do you get it administered to patients. Big difference between substance and moving to a product. And some of the products that we're producing with the new vaccines have some issues. For example, the messenger RNA vaccines are very sensitive to the cold chain. And most of these mRNA vaccines are stored at -20 or preferably at -70. Well, you don't have a lot of -70 freezers in public health nurses' offices or where vaccines are being developed. So there's going to be a lot of work done also to make sure that how we deploy the vaccines across the country will be addressed.
Okay, if there's no other questions, I'd just like to end by thanking everybody, thanking both of our speakers today for really wonderful talks, and thanks to all of you for coming. Yes, I think Jane Scholes just said it. Thanks, that was fantastic. I think that's a great way to end. Hopefully we'll see many of you in 2 weeks' time. Otherwise, have a wonderful day.
Thank you, Joel.
Thank you. Thank you very much to everyone.
Bye, everyone.